Okay, today we're going to be talking about the embryology and physiology of the human eye. Okay, we're going to start off with some embryology. Now this is basically a timeline of the most important stages in the embryological development of the eye. Now, the first morphologic evidence of eyes comes at around 22 days, where the neural folds in the future diencephalon region basically indent to form the optic sulci. So these are the neural folds. Let me visualize right here. And this is one optic sulcus and another optic sulcus. Now basically these optic sulci then expand to form the optic vesicles, which can be visualized here. Now the optic vesicles then invaginate to form the optic cup. Okay, and the lens placode, you can see it right here, invaginates to form the lens vesicle by day 32. Now the inner layer of the optic cup becomes the neural retina. You can see that right here. And the outer layer becomes the pigmented retina. Now you can also see in this second image the choroid fissure. So a choroid or choroidal fissure right here. Now the choroid basically forms due to an invagination of the inferior surface of the optic cup. Okay, and the formation of this fissure basically allows the hyaloid artery, can be visualized right here, and it later becomes the central artery of the retina, so it allows that to reach the inner chamber of the eye. Now, at around 39 days, the lens vesicle basically begins to differentiate into the lens. Now, this still continues at about 47 days. Okay, now at around day 37, the choroid fissure of the optic stalk basically closes, and that allows it to enclose the hyaloid artery and vein. Now, the hyaloid artery, as I mentioned, supplies the lens vesicle from day 32 when it forms, as well as the developing retina. Now, when the lens matures during fetal life, it basically ceases to need a blood supply, and so the portion of the hyaloid artery that crosses the vitreous body degenerates, and we'll see an image of that later on. Now, the proximal portion of the hyaloid artery becomes the central artery of the retina, which supplies blood to the retina. Now, mesenchyme surrounds the developing eyeball between the 5th and 7th weeks to form the choroid and the sclera. Now, in the 7th week of development, the mesenchyme covering the anterior surface of the lens basically splits internally to form the anterior chamber, and shortly thereafter, by about week 10, vascularization in the layer of the mesenchyme in contact with the lens forms the posterior chamber. Right there. Now, the pupillary membrane, which can be seen here, that initially separates the anterior and posterior chambers, but it breaks down, and this breakdown in early fetal life creates the pupil. Okay? So, we can also see in this image that the portion of the hyaloid artery that I mentioned earlier that traverses the vitreous body degenerates. Okay, now folds of skin from the future eyelids right here and the conjunctival sac here of the upper and the lower eyelid, they fuse together. Okay, and that happens by about the end of the 8th week, as can be seen right here. Now, by the 5th and 7th months, between those 2 months, they separate again. So here we're going to take a look at the development of a chicken eye, and this is comparable to a human eye. So if you see here, the optic vesicles basically grow in this developing chicken embryo. Now, where the vesicles come into contact with the outside wall of the embryo, a layer of cells fold, eventually becoming the neural retina. Now, at the same time, the lens begins to take shape from a thickening of the outer layer. And there we have the embryology of the eye. Okay, we're going to move on now to talk a little bit about the physiology of the eye. Okay, physiology of the eye. Now, our eyes basically respond to a part of the electromagnetic spectrum that's known as visible light. Now, this has sort of wavelength range of approximately 400 to 700 nanometers, and the retina is a highly specialized neural tissue. So, most of the cells of the retina are neurons, but among others, there are rods and cones, which are also known as photoreceptors. Now, the fovea, which is the central region of the retina, basically only has cones. Now, these require high levels of light to generate signals, and so they work best in daytime conditions. Now, cones are basically responsible for color vision and high visual resolution. So, color vision basically relies on the wavelength specificity of the cones, which respond to narrow bands of color, and also depend on sort of cortical structures, which determine the proportional response of the different types of cones. Now here you can see that there's a blue cone, red cone, and green cones, and these are basically the different types of cones that respond to different wavelengths of light. Okay, if we move on now to look at rods, now in the peripheral regions of the retina, the rods basically greatly outnumber the cones. So there are about 120 million rods, whereas there are only about 6 million cones per eye. Okay, now rods are better contrast sensors, and so they have a major role in peripheral vision. Now they're adapted to dim light, and they can't really work in brighter light. Now photopigments that are referred to as opsins are basically embedded in stacks of cell membranes in the outer segments of these photoreceptors. Okay, now these photopigments are basically able to absorb the energy of the visible spectrum. Now rhodopsin, seen here, is the light-absorbing pigment incorporated into the outer segment discs of the rods, and it contains an isomer called 11-cis-retinal, which we see here. Now, that's what it is when it's bound to opsin. Then the retinal isomerizes to an all-trans form upon the absorption of light, and it dissociates itself from the opsin. Okay, so now we move on. Basically, how do we get from there to a transmission of the signal to the visual cortex? Well, the chemical process that I mentioned just a moment ago continues. Now, rhodopsin basically binds to and activates the G protein transducin, seen here. Okay, now the activated alpha subunit of transducin binds to cyclic GMP phosphodiesterase. That's basically an enzyme. Now, phosphodiesterase activation basically triggers the hydrolysis of cyclic GMP, and as a result of this, it causes a rapid decline in the cystoplasmic cGMP levels in the outer segments of the rods. Now, a drop in the cGMP levels basically leads to the calcium channel closure in the plasma membrane, and this basically blocks the sodium and calcium influx into the cell, and so essentially the photoreceptor membrane basically hyperpolarizes, generating a direct flow of electric current along the membrane. So here we have the flow of electric current down the membrane. Now in the dark, basically the depolarized rod releases glutamate. Now, the neurotransmitter release is halted when the hyperpolarization reaches the synaptic terminal. Now, the retinal cells are grouped into two main circuits. Now, you have the vertical circuit, and that basically consists of photoreceptors, which we see right here. Okay, then we have the bipolar cells here, and the ganglions here. Now, they all make up the vertical circuit. The modulation circuit then consists of horizontal and amacrine cells. Now the synaptic terminals of the rods basically make contact with the bipolar cells, and these synapses are modulated by the horizontal cells right here, so it helps the signal to get across, basically. Now the reduced glutamate basically releases the glutamate that's released activates the bipolar cells that are hyperpolarized in the dark and respond to light increments. So this is sometimes known as on circuits. But at the same time, glutamate release also inhibits the bipolar cells that are depolarized in the dark and respond to decreases in light. These are sometimes called the off circuits. So in response to these inhibitory and excitatory signals, information is passed from the bipolar cells to the ganglion cells, seen here, and these synapses are modulated by the amacrine cells, the brown cells over here. Now, the ganglion cells, unlike other retinal neurons, do have action potentials. Now, it's the changes in light intensity and the shifting of the image over the field of vision that basically causes a change in the firing rate of the ganglion cells. So the axons of the ganglion cells basically bundle to form the optic nerves, and we just saw the optic chiasm there as the signal goes along. So that basically marks the beginning of transmission to the visual processing centers of the central nervous system. Now, activated monocular neurons then send the output to the binocular neurons, which basically begin to render the images as sort of a composite of both monocular neurons. And this mechanism is basically responsible for our stereoscopic or 3D vision. Okay, now images that are basically formed by the two eyes and transmitted to the brain's visual cortex are not quite identical, and it's this discrepancy that basically compels the brain to compare the two images, seen here as slightly different, and then to render a single integrated image, which is the final image that we see. Thank you. This concludes the talk on the embryology and physiology of the eye.